citizen, one in which everyday citizens like yourself and myself can participate. It's a way really for the lay people can play the role of citizen scientists right along the professional scientists in the efforts towards restoration. And never before has there ever been an opportunity like this to be a part of a de-extinction of a tree species. And personally, I find it one of the more interesting scientific projects of our time, as I said a moment ago, also one where a large number of everyday citizens can participate in a meaningful way. So the goal of the foundation's efforts, the foundation has been around for over 30 years, is to restore a healthy and blight-resistant American chestnut tree back into its native range. And that native range is the eastern forest of the United States. As we go through the talk tonight, I'm going to break it up into three parts that really focus around three questions. And the first one is, what makes this tree such a keystone species and such an iconic tree in our forest? So we'll take a look at the history of the tree in this ecosystem. Secondly, we'll take a look at why did it disappear so rapidly within just 2 generations. And thirdly, we'll take a look at the scientific efforts to restore it. What's the progress to date? What are the challenges? What remains to be done? So those are the three components of what we'll be talking about tonight. For those of you who may not be familiar with the various chestnut species, in the world and in the eastern forest of the U.S., there are 4 primary subspecies. And if you look at the nuts at the top left, and the American chestnut is the smallest nut of them all. Moving to the right, you then have a Chinese nut, a European, and a Japanese, the Japanese being the largest. And you really can't, though, identify a chestnut tree, be it American or any of the other subspecies, just by the nut or the nut size. You definitely need the leaves and the twigs. If you look at the leaf patterns, on the right, the one on the left is an American chestnut leaf. You'll notice how elongated it is. It has kind of a canoe shape. And you may notice that, I'm thinking this slide is not showing fully. Not a sec. Oh, why did it kick back? I don't know why it did that. I'm just going to go through till we get to where, there we are. Okay, Kara, are you seeing the full screen?
Yes, I am.
Okay.
Yep, it's good.
Okay, I didn't notice that it had kicked back. So the leaf of the American is elongated, has that canoe shape, but you also notice along the edge, the scalloped edging that is quite distinct. And that's one of the features of the American that distinguishes it from some of the other leaf patterns. The one to the right of the American is a Chinese chestnut leaf. You'll notice it's more oval rather than elongated. And along the edge, it too is scalloped, but the scalloping is more subtle and not so distinct. If you keep going to the right, you have a European leaf that is similar to the American in the sense that it's the elongated canoe shaped. It too has distinct scalloping, but not quite as large as the American. And then lastly, we have the Japanese leaf.
Excuse me, John. Hugh has a question.
Sure.
Are these nuts edible?
Yes, they're all edible. Actually, when, if you look at the European nut, which is the one third from the left, between Thanksgiving and Christmas, when you find chestnuts in the stores, it's most often the European chestnuts you'll find. Sometimes you might find the other subspecies, but it's usually the European ones. And yes, they're absolutely all edible. However, the American is the sweetest. The smallest of the nuts is the sweetest. So if we do restore the American chestnut and the nuts show up in stores again between Thanksgiving and Christmas, we'll have even sweeter nuts than we're used to. In terms of identification, I was saying that you really need a leaf and twig sample to properly identify. And if you become curious about the chestnut story and start looking for chestnuts in the woods, that actually can help the chapters in the foundation's work. And the best way to do it is if you go to the website, which I'll share at the end of the talk, the foundation's website, they have a tree locator form there, and you can take that form and fill it out and send it in with a leaf and twig sample, and they'll get back to you and tell you what you found. Maybe it's an American, might be a Chinese or European, but they'll tell you what it is. So let's move on here. In terms of finding them, the best time of year, there are really two best times. One is when the tree is in flower, which is late June, early July, noticeably later than most trees that flower. And that's one time of year. The other, of course, is in the fall, when it's producing nuts, the burrs are mature and they're opening, as you can see in this picture, one of the burrs opening with usually 2, 3, sometimes 4 nuts inside the burr. When it's in bloom in June, July, the male flowers are called catkins, and they're long, whitish, and have a strong scent, and they look a bit like pipe cleaners. And the female flowers, which eventually become burrs, they're very small at first, but by the fall, they've turned into large, green, and very spiny, and the burrs that look a little bit like green sea urchins. And you definitely don't want to pick one up barehanded. When we harvest, we wear not just gloves, but I wear insulated winter gloves when I'm harvesting in October because they're that prickly. We think about the range of the historic range of the chestnut. You can see from this chart that it ran from southern Maine all the way down through the Appalachians into Georgia, Alabama, Mississippi. But the heart of the range, the dark green area running from Tennessee up through Massachusetts, that was where the chestnut tree was most dominant as one of the hardwood trees in the forest. And they said that, the historians say that in some places, in the heart of its range, you would often see 1 out of every 4 hardwood trees would be a chestnut. In some areas, they just dominated. You'd have areas that were called chestnut hotspots that could be as large as 100 acres. It would basically be almost a pure stand of chestnuts. And chestnuts have always been known. Well, let me share this with you for a sec. There's an old myth that a squirrel could start in New York State or Massachusetts and make its way down to Tennessee or Alabama and never leave a chestnut tree. Well, being a myth, no doubt a little bit of an exaggeration, but it just gives you a sense of how dominant this hardwood tree was through the core of its range. It was always known historically to be a keystone species in our part of the world, and that means, of course, that the loss of it, they can be accurately described as they're one of the worst environmental disasters in North America in the 20th century. If you look at these two pictures, you'd think you were looking at a tree out in the along the coast of California in the redwood range. They have that kind of size to them. But these are American chestnuts in our eastern forest back prior to the blight. That the largest recorded tree was down in South Carolina or North Carolina, and it was measured at chest height. It was measured to be 17 feet across. And imagining a tree like that, you can well see why it was called the redwood of the East. So thinking about just the survival rate of chestnuts, I told you that they were not entirely extinct. They still exist in our woods, in some places more abundantly than others. But it's not it's functionally extinct is what we would say. Not officially and completely extinct or even labeled endangered, but functionally extinct. They're probably in the range from up through the eastern forest from Georgia to Maine. They're probably somewhere around 400 million is the estimate of young trees in the wild. But the great majority, maybe 85% of those trees are less than 1 inch in diameter at chest height. And they do not flower. In almost all instances, they do not produce fruit. They're only with very rare exceptions. And so we have the term functionally extinct in the sense that they're out there and in some significant number, but not fulfilling a functional role and its historic role in our ecosystem. You can see in this chart, if you look in the area of New England and in Massachusetts, some of the darkest brown areas where the surviving chestnut is the densest is around our area, Central Mass, Western Mass. So that's why I mentioned to you at the start of the talk, have any of you ever seen a chestnut, identified one in your woods walks? And if you haven't, and you start to look, and you know what you're looking for because you can identify the leaf, you might well find them. Folks could reasonably ask the question, well, if this tree is functionally extinct and there are Chinese chestnuts and other chestnuts that aren't so susceptible to this blight, well, why don't we just turn our attention to growing Chinese chestnuts and let them, play the same functional role in time that the American chestnut did? Well, here's the reason why. You can see on the left an example of an American chestnut. It's a timber tree. It grows to an enormous height, 80, 100 feet. It can form part of the dominant canopy of the forest. It's a lumber tree. Excellent for all kinds of lumber uses. And the Chinese chestnut and the Japanese chestnut, they are really been selected over time more for their orchard-like traits and shaped more like the typical apple tree. They rather squat, they broad, and the many branches lower down and not growing to the height of the American. So in terms of bringing back a tree that's a dominant hardwood, the Chinese or Japanese chestnuts simply aren't going to fill the bill. What happened there? No, I don't. Try that again. Okay. So I don't know if you're aware of it, but um, as a commodity, back in the heyday of American chestnuts, in the fall, the chestnuts would be harvested and they'd be loaded onto wagons, especially in the Appalachian Range where they were so abundant. They'd be taken to railroad depots and shipped into town, into cities where the street vendors would roast them and sell them. And in a way, you could say that the chestnuts were America's first fast food. And you might also say they were probably our last healthy fast food. It was common practice for farmers and rural folks to fatten their hogs on acorns and chestnuts. And most folks, even in towns, would let their domestic animals, their pigs and the others, just run free so that they could forage for chestnuts in the woods. Other uses, and there are a variety of other uses related in part to the rapid economic development in the 19th and early 20th century, that railroad expansion was going on at a rapid rate. There was a need for something like 9 to 12 million railroad ties around the end of the 19th century. And they were remarkably, chestnut still is remarkably rot resistant because it has a very high level of tannins. And so they were good not only for railroad ties, but for utility poles, fence rails. I actually, on my property here in Granville, there are one or two fence posts in the woods along old stone walls. They're still standing. Some of them partly they've fallen over, but I can tell by the nature of the bark that the bark is very weathered and grooved and grayish, but not rotting. And very likely those are chestnut fence rails. How long they've been there, I don't know. Another more recent story that relates to just the durability of chestnut because of its rot resistance. In Paxton, Massachusetts, back in 2008, there was a terrible ice storm, the same ice storm that devastated many parts of New England. And all the telephone poles in Paxton, they were knocked down due to the ice, except for the chestnut telephone poles that had been installed back in the 1920s. And even now, there's on Cape Cod, there's a lone telephone pole planted in 1928 that is still standing and still functional. So remarkable rot resistance. And that wasn't its only the serviceable function as a commodity. It was used in all kinds of furniture, sometimes as the backing, sometimes as a veneer. And again, it was lightweight, it was easy to work, it didn't warp, it had a beautiful grain and color. We've discovered in the floor of our old 1700s colonial house that there are boards in the living room that are chestnut boards, along with the oak ones. So not only as the source of the furniture, and other utility poles and railroad rails, it was a remarkably versatile and still is a remarkably versatile food. You can eat chestnuts raw. You can eat them raw, freshly harvested, or if you wait a few days, what happens is the starch starts to turn to sugar, so they become a little sweeter. You can cook them and roast them and boil them, puree them, add them to soups, make them as a first course or a main course, or appetizers, or even as a dessert. Matter of fact, only once in my life at an annual meeting of the Massard Island the chapter of the Chestnut Foundation. Somebody was good enough to make the chestnut ice cream, and they churned it by hand. It's the only time I've ever had it. And I will tell you, though, that it bumped into second place my previously favorite, the ice cream flavor. It was really good. But the chestnuts were not just an invaluable commodity for the human community. There's also chestnut, what's called chestnut mast. Mast is just another word for the annual production, nut production of trees that produce nuts. And a mature chestnut tree, it can produce somewhere around 10 bushels or 6,000 nuts in a season. And if you notice in this picture, there's a partridge in the lower right corner that has a chestnut in its mouth. Hares, deer, gray squirrels, chipmunks, red squirrels, turkeys, a whole variety of the woodland wildlife, they relied upon a steady and plentiful supply of chestnuts as part of their food supply. I want to show you a graph here that it gives you a sense of the role it played the pre-blight years in the eastern forest. On the left-hand side, you have a whole set of the just annual production from American chestnut and hickories, white oaks, black oaks, red oaks. And you can see altogether, there was a pretty steady flow. It varied, some years more, some years less, but there was a baseline supply of mast from these various trees for the wildlife. However, post-blight, look at the difference. Post-blight, you're just taking away one of those trees, the green American chestnut, and now you have some years where there's an abundance, and other years where there's a remarkable dearth of nut production and food for wildlife from those trees. So it's fair to say that in the pre-blight years, the carrying capacity of the eastern forest for wildlife was greater than it is now. And if we were ever to succeed in bringing back the chestnut and bringing them back in large quantity, which would take the hundreds of years to get back to where they were, well, the carrying capacity of the forest would increase again. So that brings us to the issue of what happened. And of course, what happened was the chestnut blight began to take hold. It was first identified in about 1904 in New York City by the Bronx Zoo's chief forester. His name was Herman Merkel. And this blight, they spread rapidly from New York City north, south, and westward at a rate of about 50 miles, a radius of 50 miles a year. So it spread very rapidly, and within 50 years, it had spread as far north as southern Maine, as far south as Georgia. And the estimate is that somewhere around 4 billion trees, they were wiped out by the 1950s. And it's hard for all of us to wrap our head around 4 billion, like what is that? But if you think of the geographical area of New York State, and you think about New York State being entirely wooded, that would be about 4 billion trees. So we lost an area of the eastern forest equivalent to about the size of New York State. And the result, of course, was the collapse of this keystone species, which led to a domino effect. And an effect on the ecology, effect on the economy, especially the rural economy, where chestnuts were such an important commodity to their communities. And, you know, rural families, mountain families, farmers were left with less game to eat, with less food for their domestic animals, and also the elimination of their cash crop. Remember, I said that they would ship the nuts each fall into the cities, and that money was their cash crop, and they called it their shoe money, no doubt because some of that money was used to clothe their children and themselves. So what went wrong here, what happened was this blight came along, which is actually an Asian fungus. And it was imported, no doubt, into New York City on a ship coming from Asia, maybe on Chinese chestnut, maybe Japanese chestnut, hard to tell. And the chestnut fungus is an airborne fungus. So like other funguses, it has airborne spores, and they can travel on the wind, they can be transported by birds or animals. And you can see here, see if my cursor will show up here. On the tree here to the left, you can see right in the center, there's a little splitting of the bark. And that's an area where the blight fungus has entered under the bark, gotten into the tree, and you can see the sort of orange coloring around that area. Those are the fruiting bodies of the fungus starting to spread over the bark. But the fungus is now under the bark and starting to split the bark and eat into the cambium layer of the tree. And once it encircles the trunk at any point or a branch of a tree, that branch, above that point, or the trunk above that point will die. And if you look over here on the right, you can see that the upper part of the tree is dead because there is a place, the partway up the tree, that's been infected by the blight. It has what we call a canker, and that blight canker has killed the tree above that point. So here is just another chart that shows the pace at which the blight spread from 1904 onwards. And you can see it gradually moving down the southward and moving northward. It reached southern Maine sooner than it reached areas of Georgia, Alabama, Mississippi. The black dots, they indicate counties where there are one or more surviving trees. And this is a somewhat dated chart, but you know from what I said earlier that there really are young surviving trees. Most of them root sprouts that can be found throughout the range. One of the interesting things about chestnut is that, and about the blight, is that the blight being airborne lives on the surface of the ground. It cannot survive in the ground. So the roots of chestnut trees, they are protected from this fungus. And the roots, they remain viable. And it happens that chestnuts are vigorous root sprouters. So the young trees that you occasionally may run across in the woods are, in almost all cases, sprouts from the roots of long dead trees. You don't even see the stumps anymore because they've been dead for so long, but the roots stay viable. So long as nothing else gets to the roots, they will continue to send up root sprouts for who knows how long, but they get to, this happened in my woods too. I found some root sprouts, and it gets a little exciting because the trees can get to 20, 25 feet, and you think, and it looks healthy, but you come back the next year and you realize the blight has started to take hold. So the trees were so abundant especially in the core region of the Appalachians, that there were ghost forests. You look at that picture in the upper right, and you can see in the distance, just everything is dead. And it was like a ghost forest. But these trees, even standing dead, they had a lot of value. The tanning industry took off. And flourished. New factories they put up to process the dead chestnuts. And tannins are used in not only the processing leather, but in photography, in dyeing, in certain medicines, astringents, even in clarifying wine and beer, surprisingly. And the wood was used for beams and boards, and even long-standing dead trees, because they weren't rotting, they could be used for lumber. You might have heard of wormy chestnut. Some of you would have. And wormy chestnut was just trees that were harvested, that had died. And when they were made into lumber, you would see that they had been infected by insects, but the wood was not rotting. So the wormy chestnut became something that would show up in various types of furniture. What else here might I share with you? Well, these pictures give you a sense of it. If you imagine yourself back at this time, living in an area where you depended upon the chestnut as a commodity, or you just enjoyed the chestnut trees and the chestnut hotspots, and such a long-lived tree and valuable tree. You can imagine the sense of despair that people would start to have as they watched them die all around them. It was painful, it had to be painful. And not everybody gave up hope, though. And one of them was Robert Frost. And he wrote a poem about chestnuts. And it's a very short poem. It was called Evil Tendencies Cancel. He wrote it around 1936, I think. Here's what he said. Will the blight end the chestnut? The farmers rather guess not. It keeps smoldering at the roots and sending up new shoots till another parasite shall come to end the blight. And he didn't know it, but he actually was in that last line, another parasite shall come along to end the blight. He was actually presaging one of the biological approaches that the foundation is currently exploring to restore the tree. It's not the main one, but I'll talk about it in a little bit. So with all this devastation going on, there were tremendous efforts of all types to see if they could stop the blight, if they could just find ways to protect the tree. First, there were various cultural methods that were attempted. They tried tree surgery. They tried creating forest gaps to create a barrier to the spread of the blight. And at the same time, there were other efforts going on, not back crossbreeding, but more traditional crossbreeding. They tried replacing the tree with the Asian chestnuts. All sorts of things were tried, and nothing was really making a dent in this rapid spread, 50 miles a year, of this blight. The picture here is worth making note of for a moment. Remember I said I had a fence post that is still standing along a stone wall. And if you look at the trunk, notice the gray color of the bark and the grooves in it, the deep grooves, but it's not really rotting. That is typical of a chestnut. I found a trunk like that in not just a fence post, but a trunk deep in my woods that they look just like that. And I figured, well, it's probably a chestnut. Where it touches the ground, of course, it would rot faster. But where it's elevated off the ground, the level of tannins protect it for a long time. So one of the preeminent naturalists in the early 20th century was a fellow by the name of John Meiklejohn. And he perhaps expressed better than anyone just the sadness that Americans felt back at that time with the loss of the tree. In this next slide, I'll just let you read it in silence. That's what Don said. Just a heartbreaking loss for so many people. I'm just going to pause for a moment to see if anybody has a comment, a question you want to raise before I go on to the restoration efforts. Kara, let me know if there's any question that somebody, Carolyn has a question.
Carolyn does, yeah.
Yes, go ahead.
It's not a question. I just wanted to tell you why I feel a close association with chestnut trees. And I don't remember ever seeing them, but I grew up in a house in Eastern Ohio that my grandfather had built in 1927. And he situated it behind two magnificent chestnut trees. Well, it wasn't long after he built the house that the trees got the blight and were destroyed. So what I remember the house is two huge blue spruces that they planted instead. But I've always felt very sad that I never saw the chestnut trees.
No, he never took a picture that he could share with his grandchildren.
Oh, I do have pictures of the early days of the house, but and my uncle, who also lived at that house, carved. They saved some of the wood, if not a lot, and he carved some picture frames of that wood, which are very beautiful. And I have my whole family's collection of 6 or 7 of those picture frames. So I'm reminded all the time of those beautiful chestnuts.
Well, thank you for sharing that story. That there are wonderful stories when you start to talk to people who have some connection personally through their family with the chestnut and the role that it played in the community or in a personal way in their life. They're just remarkable, the heartfelt stories. Anyone else have a comment or a question? All right, well, let's move on. And I want to take you into the restoration efforts now. And the real sort of heroes that got this underway were these two people here. They were really the force behind the formation of the American Chestnut Foundation. Dr. Burnham, they had developed, or he's the one that developed the breeding program that we currently use. And he had been a researcher in corn breeding, and he was surprised that the Department of Agriculture, they had not tried backcross breeding in their approach to the chestnut, because backcross breeding is a common crop breeding technique. And so he decided when he retired that he'd take that challenge on and see if he could apply backcross breeding to the chestnut problem. And Phil Rutter was the enthusiasm behind the project that took Burnham's research and just got things moving for the foundation. This is the logo of the foundation. It was established in 1983. And in terms of the strategies that the foundation has been applying to the restoration efforts, there are really three major components. And I'm going to talk briefly about the first one, biological controls. And then I'm going to spend a fair amount of time on back crossbreeding, because most of the chapters in the foundation are doing back crossbreeding. But equally important is to talk about the biotechnology, the arm of the strategy that the foundation has underway. So we're going to go through all three of those. Let's take the biological ones first. There are two. One is mud packing. And mud packing is simply on a individual tree. If you see there's a canker, a blight canker, one of the things you can do is create a mud pack and pack that canker. They just all the way around the trunk or on a branch, either one. They pack it in mud at that site and then wrap it in cellophane and probably use some duct tape to hold the cellophane in place above and below the canker. And why would you do that? Well, remember I said that the fungus is airborne and doesn't live in the soil. So if you pack the canker thoroughly with mud, you're basically suffocating it. It can't have any oxygen, no air, and it'll eventually die, and that canker will not continue to spread. So at the very least, you might be extending the life of the tree. So that's mud packing. But what about hypovirulence? This is, to me, although a minor approach, it's still fascinating. The concept of hypovirulence is that if you have a tree that, if you have a fungus, let's go back to what Robert Frost said. You know, if another parasite comes along that infects the blight and sickens the blight, well, maybe that would be a good thing. Well, that's actually what hypervirulence is. I'll go back to this. On the left, you have a chestnut tree that's uninfected by the blight. It isn't blight resistant. It just hasn't been exposed yet to the blight. And you see a normal bark pattern. On the right, you see an example of a tree that's infected, but it's not killing the tree. It is, the bark is disfigured, but it's a non-lethal version of the blight, because this particular blight fungus on this tree has been infected by a virus. And so it's been so sickened by the virus that the fungus can't kill the tree. And there's a very scientifically fancy name for this kind of bark. They call it cruddy bark. No kidding, that's the term we use for describing the bark of a chestnut tree that has been infected with hypovirulence. This, I think, was first identified out in a copse of chestnut trees in Wisconsin. The trees were doing well, but they didn't look normal. They weren't dying, and they didn't understand what was going on. And they studied that copse of chestnuts and discovered the virus that infected the fungus. So there is research going on regarding hypovirulence, but it isn't a major arm of the strategic efforts of the foundation. So I want to go down now to the back cross breeding, and I'm going to take a little time with this chart because I want to kind of give you a crash course in understanding how back cross breeding works with a chestnut. We'll start at the very top. And the first step is to take a pure Chinese tree and cross-pollinate it with a pure American tree. And what you get from that is what we call an F1. It's an outcross, because you're crossing one subspecies with another. That's an outcross. Let me see if I can get my cursor to show up here. There it is. So when you do that, you get this F1, and you plant those nuts in an orchard, and you wait 6 or 7 years until they're reproductively mature, and you cross them again with another pure American. And when you do that, you get your first back cross, because you've taken a tree up here. Oh, the cursor keeps disappearing. You've taken this F1 and you've crossed it back to an American. So there's your first B1 back cross. And again, you have to wait the 6 or 7 years till it's reproductively mature, cross it again with an American, and you get the next generation. And as you go along, you notice that the degree of genetic American traits keeps increasing from 50% to 75 to 7/8. And that's at the second generation back cross level. You cross that again, and you get to this level of third generation back cross, which is the first orchard that was planted here in Granville, and in fact, in the Master Island chapter, and most of the chapters up and down the East Coast, they started with third generation back cross trees that came from the research work that was being done by the foundation. So at this level, something changes though. You end up not doing another back cross, but when these trees are ready, you let them cross with each other. You let them open pollinate in the orchard. They can just pollinate with each other. And what you get in the next generation, which is the second orchard we have here in Granville, we call it a seed orchard. This is the first intercross, because you're taking the backcross trees and intercrossing them with each other, and you get this first intercross. So at this point in the Master Island chapter, what we have are orchards that are third generation backcross, and a smaller number of our seed orchards, which are the first intercross. And there's a question here, though, that I haven't addressed yet, which is the whole purpose of doing this is to breed gradually in more American traits, but to preserve the blight resistance of the Chinese tree, the genetically based blight resistance. So how does all this do that? Well, if we go back to the beginning here, these first 2 generations, at every single generation, before we do the cross, once those trees are mature enough, we inoculate every tree at every generation with the blight. We intentionally give the blight to the tree. In fact, not just the version of the blight, but actually 2 different strains of the blight. The one year and usually one the next year. And then we wait for about 9 months to 12 months, and we rate the trees to see which of those trees are showing a greater degree of blight resistance and which are showing less. We cut out the trees that are showing less resistance, and we keep as parents for the next generation only the trees showing greater signs of resistance. So that's the nature of the blight resistant, of the backcross breeding process. And there's much more complexity to it than that, but I hope it gives you at least a thumbnail sense of what the process is. So the research farm here, the Glen Price Research Farm, it was established in 1989. It's in Meadowview, Virginia, called the Meadowview Research Farm. And you have a picture of it, the part of it here. They've had tens of thousands of trees that they've planted, inoculated, culled out. They brought along generation to generation. And so the Meadowview Farm is really the bedrock of the backcross breeding program. And it's also one of the major centers for the blight research, chestnut blight research. The staff there work closely with a whole variety of collaborative partners, including colleges and universities, federal agencies, volunteer conservation groups, private industry. All of them working to assist the foundation in their goal of returning the American chestnut to its native range. You can see here just a sense of the number of chapters across the eastern seaboard, all the way up to a chapter in Maine, all the way down to Georgia. There are a few the dual state chapters like the Massford Island chapter. All these chapters, of the 17, 16 of them are doing backcross breeding work. The 17th is New York, and they're the ones that are doing the focused on biotechnology, which I'm going to talk about in just a bit. A couple more slides here. I wanted you to see just what an orchard looks like when it's in development. And as you can see, if you think about just this one orchard, and there are hundreds and hundreds and hundreds of orchards like this up and down the East Coast, you can imagine the amount of volunteer work it takes to do this. I'm a volunteer orchard manager. I have 2 orchards on my property, but there's no way that the volunteer manager is going to, manage and bring along an orchard like this. That every orchard, to be well managed, needs a volunteer crew to help the manager at times during the growing season. And you'll notice the trees are fairly close together, and that's in part because in time, as I was telling you, we will inoculate every one of these trees and then cull out the ones that don't show by our rating system sufficient resistance to the blight. So this is an example of back in July of 2018 of an inoculation process. Volunteers are being supervised by one of the science advisors to their chapter in the inoculation process, and they're going through one tree after another. They're boring a very small hole. You have a knife that's a hollow circular knife. You bore through the bark, you take out that little plug. There's a petri dish of the blight that's been grown in a medium. You take a little bit of that out, you put it into the tree, into that hole in the tree, put a little tape over it, and now you've started the process of the inoculation. You come back the 9 or 12 months later to see, well, how is that tree doing? And you can see here, let me get that cursor again, there it is. You can see right where my cursor is, hopefully it's showing on your screen, the hole in the bark. That's where the inoculation was made. And then you can see, this is probably about 9 or 12 months down the road, you can see how the blight has taken hold and started to spread up and down the bark and around the tree. And this is a tree that is not showing good blight resistance and will likely be called out in time. Here's a map, a little bit outdated, but a map that gives you some sense of just the number of the orchards in Massachusetts and Rhode Island that you'll see in your area up in the northwest corner of Massachusetts. There are several. There's one in Ashfield Hawley, there's one in Conway, in Pittsfield, Worthington, down in Stockbridge, and the one in Ashfield Hawley actually is on the property of the vice president of orchard development for the chapter. That's Brian Clark. He's done a superb job in overseeing all the orchard development in the 2 states. Down here, towards the Connecticut border is the Granville Orchard. It shows just one orchard, but actually there are 2 here on the property. So it just gives you a little sense of where the orchards are. But like with any scientific effort, the more you pursue the science, the more you begin to discover the complexity of what you're dealing with, and that the more you learn, the more you know you don't know, and that there's more to uncover. That certainly has been true with the chestnut research that's been unfolding. And one of the ways that that's playing out is that in the backcross breeding program, the initial hypothesis from initial studies of the Chinese chestnut genome is that there probably were about 8 or 9 or 10 locations on the genome that controlled for resistance. And so the way the backcross breeding program unfolded was working on that hypothesis. But as they've learned more, they realize, no, there aren't 8 or 10. There may in fact be dozens. Certainly more than a dozen of sites on the genome that control for blight resistance. So that complicates and changes the backcross breeding approach that we have underway. And we're in the midst of altering the ways that we move forward from where we are now with those seed orchards that I showed you, the 4th generation backcross orchards. But also, there's some of the good news is that along the way, there's this third arm of the foundation's effort, the biotechnological arm that the New York State chapter has been pursuing now for, I don't know, probably the last 15, 20 years at least. And this is the field of transgenics. Well, what is transgenesis? Well, it's a process where you can introduce one or more genes from one organism into an organism of a different species or subspecies. And the goal is to create a new trait in that target organism, a trait that can be passed on to its offspring. And in the case of the foundation's work, the process starts with a wheat plant. And why the wheat plant? Well, it turns out, and this is kind of remarkable in its own right, the wheat plant, first of all, is immune to the chestnut blight, immune to that fungus, and it's only one gene in the wheat genome that controls for that immunity. Well, that's kind of handy because what it's meant is that they simply have to extract that one gene, which is called the oxalate oxidase, called OXO. They extract that one gene from the genome of the chestnut, Chinese chestnut, and put it into the genome of the American chestnut. So that's what the New York State chapter has been experimenting with. And we can ask, well, what has been the result so far? What's showing up? And this is early research. They've been doing it a while, but it's slow. And here you can see some of the initial findings. On the left, you have 6 potted pure American chestnuts. And they were grown to a certain height, and they were inoculated with the blight. Same with the trees, the 6 trees in the center, but those were the transgenic trees with the OXO gene. And on the right, pure Chinese chestnuts, where they, again, grown in the same medium, in the same environment, they inoculated at the same time, and 3 of those are surviving, 3 are not. Of the wild Americans, basically it looks like none of the 6 are either alive or not doing very well. The transgenic tree looks robust. So you might say, well, oh my gosh, this is the Holy Grail. This is it. Found the solution. Let's just put these trees out in in the woods, start letting them, they propagate. But of course, it's more complicated than that. And the research is fascinating. It's ongoing. Let me explain how this OXO gene actually works with the American chestnut. This gene from the wheat plant detoxifies an acid that the fungus produces, oxalic acid. And the fungus needs that oxalic acid because the fungus lives off dead tissue. So the first thing that this acid has to do, its job, is to get in under the bark and into the cambium, the cells of the tree, kill the cells of the cambium layer, and then the fungus can start to feed off the dead tissue of the cambium layer and continue to spread with more oxalic acid taking the lead. But the OXO gene detoxifies that acid and stops the ability of the blight to proceed. So in essence, that's why the OXO gene is so valuable and has become an important piece of the research strategy of the foundation. This, however, is, this is a GMO tree. This is a transgenic tree. And in certain circles, of course, GMO, the food, GMO, the items are looked on askance and with concern. Like, what are we doing to nature? So we have to really ask, okay, this OXO gene in the wheat plant, how widespread is it? Are there other food sources or not that have the oxalic acid or the oxidase, the gene in them? And it turns out there are many, anything from beets to rhubarb to tofu, nuts, buckwheat, chocolate. They're all high oxalic foods. It's just that the chestnut doesn't have it. So although there's controversy around it, the foundation is proceeding to go through the testing process, and currently the transgenic tree is still in the laboratory. It is not being used yet with the 16 other state chapters work in back cross breeding, but the possibility is there. The key is whether it's going to pass through the federal regulatory gauntlet, which it's currently going through the EPA, the Department of Agriculture, the FDA. They all have a part in that regulatory process before the tree can be released from the laboratory. And our best guess is that from here, it's probably another 2 years before we'll know if they get the green light to take it out of the lab. And I was saying, well, this on the surface looks like the Holy Grail, but it's not, because both this transgenic arm of the strategy of the foundation and the back cross breeding arm, they both have their relative strengths and shortcomings. The back cross breeding program is rich in genetic diversity. We have used genetic material all up and down the East Coast and across the range, the historic range of the chestnut. This transgenic tree shows much higher levels of resistance than we're getting in the back cross breeding program, but it's a tree that has no genetic diversity. It's a clone. And no tree is going to survive in the wild if it's a clone. It needs genetic diversity. So how to address that? Well, one of the things that we're doing, as I wrap up here, I want you to just have a feel for the way that this science is continuing to move forward, and we're not resting on our laurels of the back cross breeding program to this point, or the transgenic tree just as a clone. We've collected in the Massword Island chapter and other chapters too, we've begun to collect wild American chestnuts from surviving pure American trees. and planting them in orchards, just pure American chestnut orchards, with the idea that we're building up the germplasm, the reserve, that if this tree, the transgenic tree, is released from the laboratory, we can begin to cross that transgenic tree with these pure Americans and begin to add some genetic diversity to the transgenic tree. That's one approach. Another, there are two more I want to mention before I wrap up. Another is to do the same thing with these advanced backcross trees, the better samples, the ones that are showing more resistance. You could take the transgenic tree and also cross them with those trees and begin to breed in genetic resistance to the transgenic tree, the genetic diversity, rather, and also begin to hopefully give the backcross trees a higher level of resistance. So it would be a win-win. In the backcross breeding program itself, and this has me particularly keen and on the edge of my seat because I've been able to be involved in it directly here in Granville. We've begun to shift away from the old breeding process and begun to focus on selecting in the lines, the very best tree in a particular line, and cross that, do a controlled pollination with another tree in another orchard from another line. That's the very best tree, a tree that's been inoculated, it still has its original trunk, it's still looking healthy, it's shown a robust response to the blight. And there aren't a lot of those trees, but they do exist. And they recently started doing that in the last couple of years, with 4 different trees, and collected the nuts, and they brought those nuts to Granville. And the couple of years, we planted 2 plots. That means we planted 300 trees in the orchard that I told you about. So some of the last trees we planted in that orchard are trees, what we call stacked resistance. We're now starting to take best by best crosses, and very careful controlled crosses, take those nuts and stack the resistance from one line with another, and begin to bring those along, and we will see what comes of that. That's a whole new approach. We have not done that before. So there are a variety of innovative efforts underway because we realize there's more complexity here to the genetic basis for blight resistance. So one last slide here. If what I've shared with you, sparks your interest at all in, this story of the chestnut, and some of you might be, interested in possibly getting involved in one way or another. There are various ways to do it. You can certainly become a member of the foundation and of the Master Island chapter. You simply go to the website of the foundation, which is acf.org, and you find a place there where you can join, make a financial contribution, or just sign up to be a member of the Master Island chapter, and you'll begin to get our notices and newsletter. You might have some fun deciding, I want to see if, I walk in the woods now and then, let me see if I can find one of these American chestnuts. You could sign up to be a volunteer for an orchard work party. And many of the orchards, like my own, have a local crew, and the ones up in the northwestern corner where you are of Massachusetts, they too need help. Or if you are a member of a group that you think would be interested in having a talk like this, free educational discussion around the chestnut story, you'd be happy to get in touch with me, and I'd be happy to share this slide presentation with others. My contact information is here. Kira also has it, so you could contact her if you wanted to get a hold of me. and the contact information here for the president of the Mass Rhode Island chapter. So that wraps up the formal tale of the chestnut's history and demise and hopes for restoration. If anybody has a comment or another question, be welcome to take it.
I just posted in the chat an article that in Delaware, a rare American chestnut was discovered at Coverdale Farm Preserve.
Mm-hmm.
Is that a big deal because it's like an original chestnut tree that survived?
Yeah, that's a really good question. If it's the article that I'm familiar with, there are, and I was talking to the lead scientist of the foundation who was up here traveling around Massachusetts not long ago, and we were talking about this. He said there are now just a handful of pure American chestnuts. They've identified genetically that they're pure American, and they show higher levels of resistance to the blight than pretty much any other the American or many of the backcross trees. They don't know quite why, but another one of the steps in this evolving scientific effort is now to take the trees and find ways to integrate them into the breeding process. I don't quite know how they're going to do that, but these, half dozen trees or so, like the one you're talking about, they are valuable genetic material for taking another step forward in breeding blight resistance. So, yeah, I didn't even know about that till this year. And it's another exciting line of investigation. So I'm glad you brought that up, Kara.
It's been posted on Facebook a couple of times already, so that's where I found it.
Okay.
So does anyone have any questions, Hugh?
No, I don't, but thank you. I am a woodworker and really appreciate this wood and its beauty as a wood surface. So thank you.
You're more than welcome. And
Thanks for coming.
yeah, thanks for joining us. And if you're walking in the woods and you think it would be fun to see if you could identify one, check out the acf.org website, pull down the tree locator and send in a sample of a leaf and a twig, and they'll let you know if in fact you found one.
I will be on my lookout. Yeah.
Okay.
And Carolyn had to leave early, but she said thank you.
Okay. Well, Kara, thank you for giving me this opportunity to share the chestnut story with some of the folks in your area. I always enjoy being able to share it. It's a hopeful story to share.
I thought it was really interesting. I didn't really know the full story, and so I really appreciate it.
Well, in some sense, none of us know the full story. It's still unfolding, right?
That's true. It's like a sad story, but with a possibly happy ending. So that's really nice.
Right. Well, we're working at it.
Well, thanks for your time tonight. Appreciate it.
You're more than welcome. Take care, and I hope your programs during Climate Preparedness Week that are coming up rapidly prove beneficial and that you get a good turnout.
Thank you. Thank you. Yeah, we'll see. So I'd like to hear how everyone else is doing on that too. So
okay. Good night now.
All right. Good night, John. Thanks so much. Bye.